Welcome back to this YouTube channel. In this video, we'll be going through ECZ questions. We're going to start from 2008 um, past paper, which is paper 2, ECZ question, up to maybe 2019. Okay, let's get started. Oh, and I'm sorry about the audio. I'm working on it soon. Everything will be working perfectly well. Okay. The diagram below shows some cells taken from a vascular tissue of a plant. Uh-huh. Then the first question A, identify the cell X and Y and structure labeled X. Okay, so as in our diagram is phloem. Phloem. You know, a phloem is composed of three types of cells: the conducting cells, the parenchyme cells, and the supportive cells. The conducting cells, also called CAV elements, are composed of columns of CAV tubes, cells that have perforations in their lateral walls, and that helps in the conduction of food throughout the plant. Then our first question A, it says that identify cell X and Y and the structure labeled Z. So cell X is companion cell, Y it is CAV tube, then Z is CAV plates. What are the functions of the cells labeled X and Y? The function of X, the function of companion cells is to supply energy to CAV tubes. Then the function of the function of Y, yeah. So the function of Y, yeah, on Y, it transports food to different parts of the plant. Okay, then we can also include the function of X, the CFA plate. This is where cells communicate with one another and dissolved food is passed through the holes during its transport through the stem. Okay. What structure in human being performs similar functions as structure Y? Blood vessels. That's the answer. D, what two conditions are necessary for osmosis to take place? Two conditions. Number one, there must be two regions separated by a selective permeable membrane. Number two, there must be a difference in concentration of solution on the two sides. Those are the conditions. Okay, we move on to question 2. Figure 2.1 shows the amount of lactic acid in the blood before, during, and after a heavy exercise. Okay, as you can see, before exercise, period of exercise, period after exercise. Then the first question: what was the level of lactic acid in the blood before the exercise? Before the exercise here, the level of lactic acid we have 20 here. So what's the difference between this point and 20 there? Wait, let me just do this. Yeah, yeah, 20 here. So the level of lactic acid in the before blood before the exercise was 18. This is 18 milligrams per, sorry, yeah, 18 milligrams per 100 centimeter cubic. That is the answer here. Then how much lactic acid was produced at 20 minutes after the exercise? At 20 minutes, this here is now 20 minutes, 60. Then at 20 minutes, it's 80. 20 minutes, it's 80 milligrams per 100 centimeter cubic of blood. Then how long did it take for lactic acid to reach its highest level after the exercise had begun? Its highest level is here. So approximately 26 minutes. 26 minutes. Okay, 26 minutes. Then let's go to the next question. Okay, question B1. What is the reason for the buildup of lactic acid in the blood? What is the reason for the buildup lactic acid in the blood? It's because of insufficient oxygen in the body, and the cells started to respire anaerobically. Then question two, what are the effects of the build-up of this lactic acid on the person? Okay, number one, muscle cramp. Number two, fatigue or tiredness. Then B3, compare the product of the process taking place during the heavy exercise to that which takes place in the yeast. Okay, the process in heavy exercise produces lactic acid, while that in yeast cells produces alcohol. Then the second one, the process in heavy exercise does not produce any gas, while that in yeast cells produces carbon dioxide. Okay, we move on to question three. Oh, oh, sorry. Before we go to question three, we're remaining with one question on question two. See, suggest two uses of the process identified in B. Okay, it is used in industry to make daily food products like cheese, yogurt, etc. Then another one is it is used in pharmaceutical industry to make medicine. Yeah. Now, question three. This is a 2010 question paper. So yeah. Figure 1.1 shows the structure of four specialized cells, A, B, C, and D. Okay. Using using the letters of the cells, identify animal cells name name them. Okay, let me just take my pen. Okay, so our animal cells is B and C. Yeah, sorry. B and C. B is red blood cells, red blood cells. Then you see it's neuron or nerve cell. Yeah, so sorry. Yeah, C it's motor neuron cell or it's nerve cell. Okay, A2. For each named animal cell in A1, state one of its characteristic features feature in animal cell 1. It has a concave shape, thin at the thin at the center and thick on its circumference. The red blood cells. Then animal cell 2. It is long. This one. It is long structured. Its length is covered with the myelin sheath, one end as a nucleus and a number of dendrons. These things. Okay. B. For cells A and D in figure 1.1, name the substance found in the cell which enables it to perform its specialized function. For A, it is the CAV plates. It, the function, it allows food to pass through these CAVs from cell to the other. From the cell to the other, yeah. Then B, A and D. Yeah, sorry. Then D, it's cellulose, huh? In a plant cell. Cellulose. Uh, cell is made up of cellulose, and the cellulose makes the cell strong and tough to protect the inner tissues and the substances of the cell. Okay, we move on to question B. Now we go to E, to C. Figure 1.2 shows an experiment on osmosis. Solution is solution F, select a the membrane. Okay, so this is H2O sugar. Okay. And the question says, from which solution in figure 1.2 will there be flow of water molecules by osmosis? Solution A or solution F? So here the answer is solution A. Huh? Solution E. Number 2. When will the flow of water molecules across the membrane stop by osmosis? Yeah. Stop by osmosis. Water molecules will stop when the solution on either side has equal concentration. When the two solutions reach equilibrium, in that case, yeah. Number 3. Suggest one reason why only water molecules can move across this membrane. Only water molecules can move across this membrane. It's because water molecules are small molecules which can be allowed to pass through selectively permeable, but large molecules are not allowed. Yeah. Okay. Question 4. Figure 2.1 shows the human digestive system. This is the human digestive system. Huh? Well, esophagus, stomach, V, W, X, anus, small intestine, pancreas, stomach, esophagus. Okay, let's get to the question. From figure 2.1, select the letters where ejection occurs. You know what ejection, huh? Ejection, it is the removal of the undigested food particles, because we have 5 processes, but others they say 6, 7, but let me just say 5. Huh? We have ingestion, digestion, um, assimilation, absorption, and ingestion. I've mixed them up. Yeah. Okay. But the first one it is ingestion, then digestion, then absorption, assimilation, then ingestion. Because when you digest, you need to absorb. After absorption has taken place, then assimilation, the usage of the absorbed food. Then ingestion, it is the removal. So here the question says that select the letters where ingestion occurs, the removal. Pancreatic juice is formed, villi are present. Okay, so where ingestion, it's X, huh? X, the anus. Then another one is what? Pancreatic juice is formed, is V in the pancreas, and villi are present, W. So here X for ingestion, this one for pancreatic, W for the presence of villi. All right. B, the stomach produces hydrochloric acid. State 2 functions of this acid in the stomach. What are the two functions of acid in the stomach? Okay, number one, hydrochloric acid. It kills some bacteria and hydrolyzes sucrose to glucose and fructose. Huh? Yeah, it kills the bacteria and it changes the environment of the stomach. Huh? Acid, it turns the environment of the stomach to be acidic, and it turns the bolus into the chyme. It activates renin, the enzyme renin, and pepsinogen into pepsin. That's the function of acid, acid in the stomach. Okay, then we go and see. Describe the roles of the liver in digestion. Okay, so the liver produces bile, which contain bile salt that emulsify fats, therefore increasing the surface area of the action of lipase. Lipase, yeah, it emulsifies. Then in assimilation, the liver converts excess glucose in the blood to glycogen, which is assimilated by the liver cells for store, storage. Yeah, for storage. Okay, 2011. The diagram below shows a plant cell which has been put in a concentrated salt solution. It has been put in a concentrated salt solution. A, B, C. Identify the parts labeled A and B. A and B. A, it's the cytoplasm. B, it's chloroplast. Okay. B1. What what happened for the cell to reach this state? What happened for the cell to reach this state? The cell was put in a concentrated solution, hence it lost its water by the process of osmosis. Okay. B2. State the term given to the cell in this condition. It's plasmolysis. It has shrink. It has what's this? Lost all the water molecules. Yeah, it is called the state is plasmolysis because it's plasmolysis is a situation in which cells lose water in a hypertonic solution. If a plant cell is placed in a hypertonic solution, the plant cell loses water and hence cell shrinks or contracts and making the protoplasm of the cell away from the cell. Yeah, that's the condition. Okay, we go to B3. How can this condition be reversed? How can this condition be reversed? Put the cell in dilute solution, pure water. That's how it can be reversed. Give one reason the structure labeled C remained remained in its natural state. Give one reason the structure C remained in its natural state. It's because it is made up of cellulose, which is stuff. Huh? It's a cell. It has cellulose. Is strong. Yeah, a tough substance. Yeah. Give two different. Am I on the right track? Okay. Give two differences between a plant cell and an animal cell. Okay. Two two differences between plant cell and animal cell. Animal cell has no cell, while plant cell has a cell. Most plant cells have chloroplast, while an animal cell has no chloroplast. All right, we move on. Yeah. All right, question six. Question six. The diagram below shows part of the respiratory system of an insect. Carbon dioxide, oxygen. Identify the parts labeled D and E. Where is E? Okay, E and D, the parts labeled. Okay, so this part, it is called D. It is the spiral. Spiral. Ah, you are going to forgive me with the pronunciation, but let me try to write it. Okay, it's spiral. I'll just spell it. Huh? Spiracle. Then E. This E is muscle cells. Muscle cells. Okay. Explain how oxygen from the atmosphere is able to reach the tissue labeled E. Oxygen from the atmosphere is able to reach the tissue labeled E. Okay, so here oxygen enters through the spiracles, which connect to the trachea. The trachea, the trachea branches into many tubes called the tracheal, which eventually reaches the tissues. Movement of abdomen and wings help air to go, yeah, to go in and out. That's how the what's this? The oxygen from the atmosphere reach the tissues. Then C, explain how the energy released from respiration in an insect is used. Okay. Movement of abdomen and wings during the process of respiration. Then you can say flying and ground movement, eating, yeah, energy release. Then we go to D. State two ways in which the respiratory system show, shown differs from that of a fish. Okay, okay. All right. So air enters insect through spiracles, huh? While in fish it enters through gills. In insects, gaseous exchange occurs across the walls of the trachea, while in fish, gaseous exchange occurs across the surfaces of the gills. All right. Question 7. The diagram, the diagram below shows a graph showing changes in volume of air during breathing in the lungs of a person. Okay, okay. Let's get to the questions. So we have stage P, stage Q, stage R, lung volume. Okay, ST. All right, A. Which letter shows the process of inspiration taking place? Which letter shows the process of inspiration taking place? You know what happens to your abdomen, huh? When you are breathing in, when you breathing in, breathing out, you know those movements, isn't it? Oh, sorry. I'm pretty sure you know, but all right, okay, let's get to our questions. Okay. So which letter shows wait, one more. Which letter shows the process of inspiration taking place? All right, so here the letters is this one. S. It's S, huh? It's S. Then two. Suggest activities taking place at stage P, Q, and R. Stage P, Q, and R here. I'm sure it's oh sorry, it's resting. Not I'm sure, but yes, here it's resting because here you are cool. You are chilled. It's resting or sitting. Then stage Q, exercise. Huh? Maybe you are running, you are doing something. Then R, it's resting again. All right. B, explain why there is an increase in air air volume during stage Q stage Q during stage Q, or here stage Q, where you are exercising. Okay, it's because the body needed more oxygen for the production of energy, which is used for exercise. That's why. We move on. Describe describe the changes which will take place in the thorax during inspiration in order to facilitate the increase in volume during stage Q where you are exercising. Okay, so they're saying that describe. Okay, so the diaphragm contracts and moves downwards, flattens. Yeah, then external in intercostal muscles contract and internal intercostal muscles relax. Then the rib cage move upwards and outwards. Volume of the lungs increases while pressure in the lungs decreases. The air is drawn in. All right. Question 8. Figure 5.1 and figure 5.2 show the cross section through two different portions of the human alimentary canal. Human alimentary canal. Okay. From which part of the alimentary canal where these cross sections are taken? Okay, so for figure 5.1 is the small intestine, the ileum. Figure 5.2 is the duodenum. That's the duodenum. Okay. A2. Give reason for your answers in A1 above. Okay. Figure 1. Figure 5.1. It's because it has projection. It has projection in the inner lining called villi, called villi or villas. Then figure 5.2, because it has a duct which opens into it, which opens into it. Yeah. All right. Then we move on. We move on. 8B. Where is B? Explain. Yeah. Explain the role played by B in the alimentary canal. It contains longitudinal and circular muscles that contract and relax during peristalsis and keep the food moving on its way through the digestive system. Then C, name two juices passing through tube C. Name two juices passing through tube C. Okay, tube C, that's one. It's bile and pancreatic juice. Okay, 2014 question. The diagram below shows the movement of dissolved substances in the digestive system and some associated parts. Okay, bloodstream. Let's Google. glycogen, lactic acid, okay, ileum tissue. All right, let's get to the question. Name the structures labeled I and J. I, I and J. Okay, the glycogen. You know that these things of these glycogen occurs in the what this liver and lactic acid or glucose into lactic acid. It's in the muscles, the muscle tissue. So I I is liver, then J this one is a muscle tissue. Okay, then we move on. B1. Name one product of digestion in the ileum which is not transported by blood. One product in the ileum which is not transported by blood. It is fatty acid and glycerol. Fatty acid, fatty acid and glycerol. Okay. Name the enzyme in the ileum whose activity produces glucose. Whose activity produces glucose. It's maltase and lactase. Maltase and lactase. All right. We're on name. Okay. See, substance K. Substance K, when broken down, produces a waste substance and glycogen. Identify substance K. Where's substance K? Okay, this substance K. All right. Identify substance K. Okay. So substance K, it's amino acid. It's amino acid. That's substance K. Suggest two uses of substance K in the body. Suggest two uses of excuse me. Excuse me. I'm sorry. Sorry for that. Okay. Suggest two uses of substance K in the body. Two uses of amino acid. It is used in growth and repair of body tissues, and it is used for making body chemicals such as hormones, enzymes, antibodies, etc. Okay. Um, deep state state under what conditions the product in structure J is produced. Um, it is produced when there is not enough oxygen in the body due to exercise, like running. This substance structure J is produced. Yeah, the lactic acid when you're doing exercise, when you're stretch, strenuous exercise, it's going to be produced. Okay, 2015. Figure 1.1 and figure 1.2 show the structure of specialized cells in plants. We have these ones. Okay. Identify cells labeled A and B. Identify cells labeled A, labeled A and B. So this A of A, B, C. All right. So A is the epi, epidermal cell. B is the guard cell. Yeah, B is the guard cell. Then identify the parts labeled C and D. C and D. C is the chloroplast. Then D is cytoplasm. Huh? D is the cytoplasm. Okay, B1. Which figure show a closed stomata. Which figure shows a closed stomata? Figure 1.1. This one is closed, this one is open. Yeah. Then explain how the opening of the stomata is brought about. Okay. During the day, potassium concentration in a vacuum of gut cells increases. This this lowers the water contents in the cell sap, which led to the flow of water from epidermal cells to the gut cells. Yeah, then this inflow of water rises the tangle pressure inside the gut cells. Huh? Then the pressure causes the gut cells to cave in such a way that the stoma between open between the open. That's how the opening of the stomata is brought about. And the other thing you can say that yeah, you can say stomatopods open when gut cells enlarge as a result of water absorption and close when gut cells shrink. All right, we go to the next question. All right, we are done with here now. 11. We're on 11. Okay. Figure 2.1 shows the section through a human tooth. FGHI 2.1. The question. State the names of the parts labeled F and G. State the names of the parts labeled F and G. So F is the enamel. G is the dentine. Yeah. Explain uh, explain the functions of the parts labeled H and I in figure 2.1. The functions of the part labeled H and I. Okay. H H H this is a space within the dentine which is made up of tooth producing cells, nerves, and blood vessels. The nerves makes the tooth sensitive to stimuli such as temperature, pH, and pressure. The blood vessels supply the tooth with food and oxygen. I This is a bone-like tissue with fibers that anchor the tooth into the jawbone. That's the function of eye. Then explain how tooth decay brought about. How tooth decay is brought about. Okay, tooth decay is brought about when sweet food particles stick in between the teeth for so long and form a plug. Huh? The bacteria starts to feed on the plug and respire anaerobically, producing an organic acid. This acid dissolves or corrodes the enamel of teeth, forming a cavity in the teeth. When the cavity reaches the dentine, the tooth starts getting painful. The pain increases further when the cavity reaches the pulp cavity, and this is called tooth decay. All right, then B2 suggests two ways of preventing tooth decay. Number one, brushing teeth with fluoride, fluoride toothpaste after every meal. The second one, avoiding intake of sugary food. Okay, then we go on figure 3.1 shows a cassava plant with root tubers. With root tubers. Okay, let me just do this. Yeah. with root tubers. Then the first question, A1, which which common food nutrient is stored in the root tuber? Which common food nutrient is stored in the root tuber? It's starch or carbohydrate or glucose. Number two, state where this food in the root tuber was manufactured. It it is in the leaves. It is in the leaves, yeah. Three, explain how this food manufactured in A to above found itself in storage from form in the tube. Oh, excuse me. Um, Sorry for that. Yeah, I'm sorry for that. Someone was just calling. Okay. We're on three. Explain how the food manufactured in A to above found itself in storage form in the tuber. Storage form in the tuber. Okay. So when glucose is manufactured in the leaves, it is combined or converted to sucrose, which is transported from the leaves to the tuber via phloem tube by the process of translocation. In the tube, sucrose combined forming starch and stored in that form. Then B1, state the nutrient in the soil which is necessary for photosynthesis in the soil, the nutrient which is necessary for photosynthesis. It is H2O. It is water. Yeah. Okay. So here, yeah, the what's this nutrient? It is water or mag or magnesium. Magnesium you can say heavy nitrogen. Yeah, but magnesium and H2O mostly. Okay. Then suggest how this this nutrient is taken up from the soil to the leaves in the plant in figure 3.1. Okay, so let me say water in the soil is absorbed by the root hair by the process of osmosis. It is taken up through the xylem vessels, which sorry, with the help of transpiration poo. Yeah. Then root pressure and guttation, it will be helping the what's this the water to be absorbed until it reaches the leaves. Okay. Then this sorry. Not then this how this nutrient is taken up. All right. I've just explained that. We go on to 2015. Figure 3.1, the life cycle of a housefly. 3.1 life cycle housefly. We have GHI. Identify the stages labeled G, G and H. Okay, so G and egg. H, the second insert stage, yeah? It's the second like lava stage. The second lava stage. Explain the changes taking place during stage I. The changes taking place during stage I. It is increasing greatly in size and it is reducing in production of predatory maggots that feed on other maggots. Um, it is reducing in feeding on rotting materials by sucking the nutrients and moving um, using fat on the lower side of their body, huh? Of their bodies. That's the worst stage. That's the changes taking place during that stage. Then B1, which stage into the 3.1 would be the most effective for eradicating houseflies? Okay, so the pupa stage, because it's inactive and not moving, just stationary. Hence, it is like easy to kill it, huh? Something which is not moving, like it's like lack of, let me say, lack of a better term. Let me say it's stagnant, huh? It is it is not moving. So you can kill it by spraying with insecticides. Insecticides, yeah, at that stage of pupa. It is also not being able to reproduce, so it is very easy to eradicate it. All right. Then bit two. Using a named example, describe the role of a housefly in disease transmission. Houseflies are vectors for pathogens that cause cholera, dysentery, and typhoid. Okay, we move on. 14. Figure 4.1 shows the growth of Milka on Shima. Milka, I don't know if it's Milka Mark or on Shima. Okay. Identify structure J in the diagram above. Okay. So the structure J, it's sporangium. Sporangium. Then explain how structure K obtains nutrient from Shima. K, structure K obtains nutrient from Shima. Okay, so the mucus, it's the mucus. The mucus releases enzymes from its hyphae into Shima. These enzymes hydrolyze the Shima into glucose as the end product, and the structure K, the stolon absorbs this end product, absorbs this end product. Okay, so then that's for part A2. And the structure K, it's rhizoid, huh? Not stolon, rhizoid. This is the stolon. Okay, um, C or B. The Nishima was flooded with iodine solution. State color changes that could occur at L and at M. Okay. At L, color changes to blue or black. At L, starch starch is present there. Then at M, no color changes. It remains yellowish or brown at K. Okay. See, give give reasons for the color changes observed in B above. The color changes observed in B above. At L, it is because of the presence of starch, and at M, it is because of the absence of starch, because starch has been converted to glucose by the enzymes secreted by the um, mucus. Milk. All right, 2016. Figure Figure 1.0 shows the setup of the setup to an experiment used to investigate the effect of biological catalysts on the decomposition of hydrogen peroxide to produce oxygen. All right, potato juice, hydrogen peroxide solution. Okay, three experiments were carried out under different conditions as shown in the table below. Experiment number one. All right, boiled raw. Okay, let's get to our question. Um, what is the general name given to a biological catalyst? The answer here is enzyme. Enzyme. Give a reason for boiling the potato discs in experiment two. In experiment two, um, to denature or kill enzymes, catalysts in it. That's the reason. Then explain the difference in results obtained between experiment one and experiment three. Between experiment one and experiment three. Okay. In experiment, okay, let me say something in experiment one. The the volume of oxygen produced is very small. When yeah. The volume of oxygen produced is very small. This is because almost all the enzymes in the potato dish are denatured. Hence, very, very, very like lack of a better term, minor enzymes to convert hydrogen peroxide to oxygen and water. In experiment three, the volume of oxygen produced is very much. This is because all the enzymes in the potato dish converting converting hydrogen peroxide, producing oxygen and water, producing oxygen and water. Okay. Number B. Predict what yeah, predict what would be the result of number one. Increase the number of potato discs in 5 centimeter cubic of hydrogen peroxide peroxide. Okay, for B1, predict of hydrogen peroxide. Okay, so um, more oxygen gas will be produced with very short time. This is because enzyme concentration has been increased. Then B2, putting the mixture of 5 million cubic of hydrogen peroxide with potato disc. Okay, so as the temperature increases, also the production of oxygen gas increases. This is because reactivity of enzymes work well at optimum temperature. See, suggests two other characteristics of a biological catalyst not demonstrated in figure E or in A and B of this question. Okay, number one, enzymes are specific in nature. They have a specific substrate to catalyze. Two, their activity is affected by pH, meaning some enzymes work well in acidic medium, while others they don't work well in an acidic medium. All right. 16, question 16. Figure 2.0 shows cells taken from a vascular tissue in a plant. Identify the specialized cells in figure 2.0. Specialized cells. Okay, so that's we have A, B, C, D, E. A, B. Okay, so A it's phloem cell. Then letter B. Wait, let me just move this over here. Okay, yeah, okay, sorry. So identify the specialized cell into the 2.0. It is the phloem cell. It is the phloem cell. Then name name one feature in figure 2.0 which enables you to identify the specialized cell in A above. Name one feature. All right, so using that name one feature. So we really just attempt to do this one. Okay, so as in for number two, the feature is the companion cell or the cellular plate. Then which letter on figure 2.0 corresponds to the following structure? Okay, nucleus, cytoplasm. A is for nucleus. Then cytoplasm is the cytoplasm is it's D, huh? I bet D, yeah. D is for the cytoplasm. Okay, then B, we on B, yeah, B. Give two functions of the cell labeled B. The function of the cell labeled B. Cell labeled B that was supposed to be, is it the companion cell? Yeah. Is it the cell or the companion? Okay, this structure is it's not that clear. Okay, so I don't want to give you false information, but I'll actually just end here. Thanks for watching. Please don't forget to subscribe and leave a comment. If you have observed any mistake, just comment in the comment section. Otherwise, thank you. I'll be back with another edition.